So the third part of the video on transcription factor and transcriptional control part 1 is actually on regulation at transcription elongation. So I think I mentioned this before in last week video, one of the video anyway, on basically elongation is actually a complex process. So it's, well, as the name says, okay, it needs to be elongated, so it will be elongated as long as the gene needs to be transcribed. So it involves two stages, early elongation and productive elongation. So after initiation, so polymerase II will start the iterative incorporation of nucleotides at the 3' end of the nascent RNA transcript as the RNA transcript goes along. So elongation by polymerase II is not always uniform throughout a gene because, of course, nothing is perfect. For example, in metazoans, polymerase II transcribes the initial about 20 to 60 nucleotide of RNA and then pauses. So progress beyond this point is rate limiting for approximately half of all active Drosophila and mammalian genes, and the escape from the pause into productive elongation is often highly regulated. So what is early elongation state? So it is the transition of polymerase II between promoter escape and fully productive elongation, which is often accompanied by an intermediate step of promoter proximal pausing. So there is a pause state, okay, during the elongation state. So this transition of initiation, basically the polymerase II, okay, will pause, okay, which occurs at nascent RNA within a length of around 10 nucleotide, okay, and extended to around 20 to 60 nucleotide, okay, depends on the types of organism, and this was measured most precisely in Drosophila. In others, basically it hasn't been published. So the bulk of this pause polymerase II in mammalian genes occurs in a similar position or state to be. So when the RNA chain is less than 10 nucleotides long, the polymerase II is still considered to be within the initiation state. So it is associated with TF2B and can be terminated prematurely, which is referred to as abortive initiation, which may provide checkpoints for promoter control. So once the RNA grows longer than 12 nucleotides and TF2B is ejected, the polymerase II elongation complex becomes stably engaged, and of course it is now in the early elongation stage. So early elongation is actually not a simple smooth transition of polymerase II, but of course it involves critically regulatory steps, as we will see. So these are some of the factors named that is involved in transcriptional elongation, as you can see. So don't worry. Again, most of the time students will ask, do we need to remember this? No. Because the thing is, it's actually a complex mechanism, and if we were to study the transcription in eukaryote as a whole in one course, then probably yes, okay. But at this moment, actually, we do not need to remember each and one of them, but just know that there are lots of factors involved during transcriptional elongation, as you can see here. Alright, so let's have a look. Okay, so this, of course, everything from this topic is actually taken from journals. So as you can see, these are also taken from some journals on the structure of the promoter and polymerase II before and during early elongation. So in the A1, the ones on your top left, the promoter is in a closed configuration, and of course, this is occupied by nucleosome. So we know that nucleosome positions are, of course, the distance between the diet axis and the transcription start site, in base pair. And basically, what happens is that, when it is in an open promoter position, basically it is unbound by a nucleosome and of course occupied by general transcription factor. So this would mostly more or less gets or tells you that it is ready to start off transcription. So in the C, it's basically the pre-initiation complex, PIC, not a person in charge, but pre-initiation complex. So it is composed of the promoter DNA, polymerase II, Tata box binding protein, your TBP, and your TF2D and other general transcription factors, those involved to make the pre-initiation complex. So now, part of the TF2D subunit is actually removed to make it easier to visualize organization of other general transcription factors, as you can see over here, but probably not so, because actually TF2D is quite a big structure. So in the D is promoter proximal pausing. So polymerase II is paused between the promoter and the first nucleosome. So pausing position is at around 40 base pair, 40 nucleotide from the transcription start site, and this is typical in Drosophila promoters. In mammalians, it has been thought to be in various position. So what happens is that, okay, polymerase 2 bound NELF or negative elongation factor, DRB sensitivity inducing factor, DSIF, and TF2S. Okay, so as you can see, all of them are here. So TF2D may or may not be present at the promoter depending on the residency in the reinitiation scaffold. So this is of course based on the crystal and cryo-electron microscope structure. Alright, so what do we know about the promoter proximal pausing? So this is the molecular models of it. After initiation, the early elongation polymerase 2 meets its first roadblock and pauses. So that is why it's called pausing. So the identity of this roadblock is very important because it provides the strongest rate-limiting step in the course of polymerase 2 elongation along the gene. So various in vitro studies have identified many different components that make up this stop sign in various organisms. So the mechanisms for establishing the pause in vivo can be summarized into three models. They are not mutually exclusive, which means that it could vary, okay, from one organism to another and depending on the condition. Okay, we have kinetic model, we have barrier model, and we have interaction model. So the promoter region around the transcription start site contains several conserved sequence motifs, which is recognized by the general transcription factors and polymerase 2, as you can see over here. Now, models of pausing for different promoters that involve distinct inputs and the mechanism for proximal pausing, proximal promoter-driven pausing, and for distal nucleosome-driven pausing basically varies. And the structure is, as you can see again down below. So for a promoter with a stronger DNA element, and it would depend on the sequence, so the interaction model dominates the energy landscape, and the output is position proximal, driven by the promoter factors, as you can see over here. But okay, in the D structure, okay, for a promoter with a weaker DNA element, nucleosome barrier defines the energy landscape more dominantly, and the result is the pausing position distally driven by the nucleosome. Okay, so one is promoter driven, one is nucleosome driven. Now, let's have a look, okay, at the three models: kinetic, barrier, and interaction, and what we know on the promoter DNA elements, okay, and also of the promoter driven pause and nucleosome driven pause. So as you can see, okay, this is the pausing or proximal pausing of polymerase 2 conditions that could occur. So the descriptions, okay, basically are explained here. So I'm not going to repeat it in this video. You can just read about it in your notes. What happens, okay, is that as you can see, okay, these are the two most common types of pausing, proximal pausing that occurs, okay, either by promoter driven or nucleosome driven, and it is mostly, okay, based either on kinetic model, barrier model, or interaction model. So for these two, okay, you can actually guess, okay, which one of the model they are from. Okay, can you guess which one of them they are from? Okay, for promoter driven pause and also nucleosome driven pause based on the structure. Okay, so if you could, then good for you. If not, then probably, okay, you need to look at how the whole structure is basically positioned to determine what models they are. Now, what would be the fate of pause polymerase 2? So promoter proximal pause polymerase 2 can resume the incorporation of nucleotide triphospholate, the addition of the nucleotides, and enter productive elongation. So it has also been shown that vast majority of pause genes have detectable levels of elongating polymerase 2 in the gene bodies. So measuring the ratio between the pause and elongating polymerase 2 makes it, of course, possible to estimate how frequently polymerase 2 escapes into productive elongation and assess the activity of the promoter. But pause polymerase 2 may also terminate instead of resuming elongation. So in eukaryotic cells, polymerase 2 that has stalled at DNA damage sites can be removed by the ubiquitination-mediated degradation pathway during transcription coupled nucleotide excision repair. Now, how does it move, okay, from proximal pausing to productive elongation? So this is referred to as escape into productive elongation, which means that it escaped from being terminated. So productive elongation is defined as the efficient progression of polymerase 2 through the gene body, and the escape into productive elongation refers to the transition of the pause polymerase 2 into productive mode. So this is where, okay, your gene is being made, okay, your mRNA is being made, and of course, okay, that would determine how it's going to be expressed. So while polymerase II is resident in the promoter proximal region, two important modifications to the major component of the transcription elongation complex takes place. So the first one is the 5' capping of the nascent RNA. Second, phosphorylation of the post-polymerase II complex. This is of course mediated by protein kinase complex PTFB, which also phosphorylates the pausing factors TSIF and also NELF. So at this stage, what happens that polymerase II and its associated nascent RNA are modified so as to allow recruitment of additional elongation, which means that it would allow or enable the addition of nucleotides. So these RNA processing factors help polymerase overcome further barriers in the gene body and couple transcription and of course RNA processing. So this is from the post-polymerase II. So the recruitment of active PTFB is a critical step in to control the gene expression. Of course, it's regulated at multiple stages. So in human cells, so since we are all human, the free form of PTFB is sequestered by 7SK SNP. So small nuclear ribonucleoprotein for those of you who have forgotten. So this is of course is in an inactive state. Now, upon activation, PTFB is released, as you can see over here, PTFB is released from sequestration and recruited to the promoter in a variety of ways, as you can see. So it is recruited to be either forming a complex here, or basically it is going to be more or less on its own, or basically with addition of some other factors, and of course it may also be accompanied by a mediator. Now, after completion of its function, the PTFB can be sequestered again by the 7SK SNP, but in some highly active gene, usually it remains bound with the elongating polymerase II in super elongation complexes, as is CS, as you can see over here, elongation. So what happens is that how to escape from the proximal pausing into productive elongation. So once polymerase II makes the transition from the pause state into productive elongation, it will then of course progress through the gene body. So in the gene body, polymerase II faces other barriers and requires additional elongation factors to overcome them. So that means that it's not a smooth journey for them. They have to overcome a lot of things before your gene could be transcribed, so you should appreciate your genes. So finally, polymerase II transcribes through the 3' end of the gene, where your nascent RNA will be cleaved and polyadenylation define the end of the mRNA transcript. So addition of the poly A will be added. The actual release of a transcribed polymerase II molecule from DNA usually takes place on average around 8 kb downstream of the 3' end. This is in human cells. So in Drosophila, it's actually much lesser than that. And processing of the 3' end of your nascent RNA and polymerase II termination adds an extra dimension to the co-transcriptional and post-transcriptional control of gene expression. And this could be regulated and could also determine how your gene could be expressed. Alright, so basically those are more or less the pre-introduction of transcriptional control. We will go further for the second part next week, and we shall see what else is there in store for you guys.